Yes, चलो बात करें. आज के टॉपिक का I think it will be a little bit more interesting. ठीक है? So we are starting with immunology. Start करके immunology अपना की पढ़ना है. Antigens पढ़ना है. So what are antigens? What are antigens? Basically what are they?
They are the proteins present on the surface of the organism.
They are proteins. So antigens are some kind of proteins that why do they call antigen? Because they produce some kind of, they generate some kind of proteins against them. Okay, that is why it is the simple meaning of antigen. So what do they produce? They produce antibody. So today's class is about antibody. This is going to be a very short class, but it's very important because this might come as a short question for you. So there are total 4 kinds of proteins in plasma. One is albumin, another is globulin. Globulin, amongst them, gamma globulins are the main constituents of antibodies. Okay, and alpha, beta, those proteins are very smaller ones. There is a test called electrophoresis. Electrophoresis means when we put a plasma and we put a current, electric current through it, so we get sometimes this kind of graphs in electrophoresis. X-axis and y-axis. And if you are talking, imagine the plasma and we are depositing a electrophoresis graph for electrophoresis. You are putting one electric current through a plasma. The machine is interpreting whichever protein is the highest, will give the highest peak. So first the peak we get the albumin. After that we get the alpha and beta globulins. After that we get the gamma globulins. So antibodies they come under this gamma globulin structure. Okay. So antibodies are constituted of gamma globulins produced by plasma cells. What are plasma cells?
Okay.
The activity for what are plasma cells?
Modified B lymphocytes. Modified B lymphocytes.
Oh, modified B.
B lymphocyte after.
B lymphocyte. Very good. So it is used by our immune system to identify and neutralize. So basically, what we need to study is we will study about this structure. I remember when I was studying for NEET preparation, আমরা তখন ড্যান্স ছিল। তো ড্যান্সে কেউ তোর সবাই ম্যারাথন ক্লাস করছিস একবার ম্যারাথন করছিস কিন্তু সো তখন আমাদের ড্যান্স ছিল। আমাদের ড্যান্সে মাইক্রোবায়োলজি পড়াতো সাইন্স ভালো লাগতো। যে মাইক্রোবায়োলজি পড়াতো যে পড়াতো সে দেখাচ্ছিল এরকম না আমি শুধু লোকটা মুখটা মনে আছে যে এরকম করে কিছু একটা পড়াচ্ছে লেখা এমনি করতে এসে বুঝতে পেরেছি যে ওটা অ্যান্টিবডি পড়াচ্ছে ওকে the structure of antibody not a single type in generally antigen we mark antigen like this when there is a point which get attached to a antigen. Antigen is written as a Y. So this is called the place where the antigen gets attached to the antibody is called the epitope. And here wherever this is called the epitope. And this is called the paratope. Okay. Next point will come. So this is the normal, like layman structure of a, if we consider layman structure of an antibody. So this is the one heavy chain, this is another heavy chain. Two heavy chains are bound by disulfide bond. Whatever point I am telling you, that much if you write, it's enough for you. I'm not going to slide by slide. I'm just telling you this. Okay. And both the light chains. This is the light chain. This is the light chain. Both the light chains are bound by also disulfide bond. So the antibody structure holds together by disulfide bond. Okay. So antibodies are also called immunoglobulins. Okay. And antibody recognizes a unique molecule. So a particular antibody's paratope recognizes an antigen's epitope. Okay. So there is a particular epitope, a particular paratope for a particular epitope. So what happens? So the molecule of the last one, harmful agent called antigen via variable regions. This is the history at 1940. The fact was recognized. So there is an antigen, an antibody against each antigen. So next point is important antigen bivalency. What is the valency of an antigen? So see, for to know what is called valency. These are the very basic things you need to know. What is valency? Valency of an antibody. So I told you there is a place, particular place where antigens get attached. So each antibody has two places. One is ABB, and FC. Minimum valency can be 2. There can be valency up to 10. In which group of antibody can
IgM.
Okay, but IgM cannot valence
Pentamer.
is like this. Total parts are so it should be 5 into 10. But normally there should be 10, but IgM is pentamer, not 10 के ये बोले की बोले I know
Deca.
very good, you know better. So 10 valence is our kotha, but 10 valence is not, that is pentamer. And double valence का कौन antigen है? IgG. That only IgG, IgD and IgD. They are bivalent. IgA has double valency, means it has two molecules of antibodies together. Okay, and IgM is made up of total 5 molecules. Okay. So this is the structure that I was talking about. So this is the region which I have drawn. These two regions means this region and this region. Okay. This is VH and this is called VL. VH and VL. So this part is called variable. By variable, they can constantly change their molecular arrangements. Okay, so that according to the antigen, they can In exam, if you just can draw this structure, it means you know about antibody. Okay. So what are the types of antibodies? Composed of two identical heavy polypeptide chains and two identical light chains. So what are the types of light chains? There are two types of light chains. One is kappa, another is lambda. And based on different antibodies, different types of antibodies, there are four types of heavy chains. What are they? Alpha, gamma, delta, epsilon and mu. 5 types total. So alpha is in, naturally, alpha is in IgA. Gamma is in IgG. Delta we find in IgD. Epsilon, E and mu is in IgA. Okay. So each heavy chain has two regions, one constant and one variable. Alpha and gamma chains contains approximately 450 amino acids. Okay. While mu and epsilon chains have 550 amino acids. That's why IgE and IgA, they are bigger molecules. Which antibody amongst them can cross the placenta, blood-brain barrier? IgG. Okay. So when a child is born, he or she already has mother's IgG. So in generally, IgG already persists in the child's body. So আমরা কি বলি বাচ্চা 2 বছর অবধি না হওয়া অবধি অ্যান্টিবায়োটিক্স দিও না, because let their own antibodies work. Okay. So light chain, I told you about, there are two types of light chain, kappa and lambda. All antibodies have two kinds of light chains. দুটোই থাকে সব অ্যান্টিবডিজ kappa and lambda. A light chain has two successive domains, one constant and variable. Approximate length is 212 and 262 or 217 amino acids. These things are important. These things you have to get it from the slides. Number of amino acid present in the, these are all polypeptide chains. The number of amino acids in the light chain is 211 and 217. This is the exact picture what I have. So IgG, this makes up 80% of serum's antibody load. Okay, 80% of antibody are IgG, and their half-life is 7 to 22. What is half-life?
The time in which the antibody count becomes half of its initial level.
Half of the exactly. So IgG is a monomer. I told you it's a monomer, it's a single molecule, and it has two epitope bindings. So this is the structure of IgG. So this has two epitopes. This is one epitope and this is one epitope. Okay, epitope binding site, two epitope binding. Okay, and this it has two paratops. Okay, and this is the only class of antibody can cross the placenta and enter the fetal circulation. Function is immunity to the newborn and neutralization of toxins, and IgG binds to Fc receptor of the phagocytes. This Fc and FAB receptors, we will talk about it in your complement classes, complement system classes. Complement পড়ানো হয়েছে তো? Complementary class এ পড়ানো হবে। Fc and FAB regions required. Okay. So next is IgA. IgA consists of 13% of the antibody load in a patient serum or plasma. Okay, and the half-life is 5 days. So which antibody? Imagine if you are getting a viral infection. If you are getting dengue infection, first we NS1 antigen. NS1 antigen. Okay. We first detect the antigen. The antigen is in early stages. Whenever a patient is coming with a fever or anything, we first try to detect whether the antigen is present or not. If the antigen is present, means the patient is in active infection. Patient has active infection. Whenever we start getting antibodies, that means the body started reacting to the antigen. First antigen we get is
IgG, IgA,
IgA.
in active infection we get IgM, and in chronic infection we get IgG. Okay, so in dengue, if we want to detect if it's a dengue fever or not, first we send for NS1, then IgG and IgA. Nowadays there is a rapid card, okay, on their rapid card, activate jole rapid card, but the IgG, IgM card kore. It's not supposed to happen like that. Biology class is introduction. What's that? One and two start. Okay. So, IgM is the first immunoglobulin class produced in a primary response of an antigen. So, it is a pentamer and 10 epitope binding site. মানে হিসাব মতন 10 epitope binding site হওয়ার কথা, but there are always 5 active, not 10. Okay. Functions কি হচ্ছে? Activation of classical pathway. This classical pathway is a part of complement system. There are two pathways, classical and alternative pathway. So the classical pathway, it starts with IgA, okay, and defends against multivalent antigens. Means there are some antigens which has different kinds of epitopes. That's why IgA is the first one to enter in the system and take the action against the antigen, because it has different kind of paratopes. Means if a very potent virus is coming, a potent bacteria, any kind of antigen is coming with various sites from where it can produce infection, IgM is the first one that can protect it. Okay. And it acts as a opsonate. What is opsonate?
antigen antigen.
This is a class also, na brother? Pathology में पढ़ाना है ना opsonate?
Yeah, it helps to puncture the bacterial membrane.
So next is the IgA. IgA, why it is important? Because it is, although it is only 6% in the serum and a half-life of 5 days, and it is 4 epitopes. That means IgA molecule is like this. It is like it has total 4 epitopes. Okay. So basically what IgA does, it gives the mucosal protections. So in our mouth, wherever mucus, in the gut, wherever there are mucosal membrane, there is IgA present. Okay. So, and another thing, a baby, it gets IgG from the mother directly. After birth, why do you tell for exclusive breastfeeding? To grow baby's immunity. In the colostrum and the milk, there is IgA present. Okay. IgA, that gets delivered from the mother to the baby through breast milk. That's why we tell for the exclusive breastfeeding. So this is the secondary antibody, and nowadays, so বেশি যে ইনফেকশনস তো ইনফেকশন এখন চারিদিকে হচ্ছে, বোলপুরে তো খুবই হচ্ছে, H1N1 খুব হচ্ছে, so it gives protection from influenza, okay? And in That's why baby needs to drink the mother nowadays after C-section they insist on having fed their child the first milk. First milk means the cholesterol. Cholesterol has the maximum quantity of IgA. But nowadays C-section ratio normal delivery common until and unless normal delivery also the place the whole system gets to the end. In season what we are doing? We are terminating it at 36 weeks or 37 weeks or 38 weeks. We are not carrying up to the term. So the cholesterol it starts time to take. But the baby it cannot wait for 72 hours to make the cholesterol start producing. So we have to give the baby formula feed or something. But the mother will keep on insisting बच्चे को खिलाना है मैदा को खिलाना है ये वो कहते हैं ठीक है This is the thing যার জন্য আজকাল বাচ্চাদেরকে এত অ্যান্টিবায়োটিক দিতে হয় কিছুদিন আগে একটা আউটবাস্ট হয়েছিল আউটব্রেক হয়েছিল মিজলস এর বাচ্চাদের খুব ইনস্পায়ার্ড অফ গেটিং ভ্যাকসিন মিজলস ভ্যাকসিন কত মাসে পায় এনি আইডিয়া MMR
6 months. 8 months.
Weeks na baba. 9 months again. Weeks se kichu na. Weeks se onek aage paye. Aage pentagonal doctor. This outbreak হচ্ছিল 1-2 বছরের বাচ্চাগুলোর মধ্যে. Because nowadays 98% of babies are cesarean born. So they did not get the proper cholesterol or the proper breastfeeding. That's why their immunity didn't get built up. Okay. So the next one is IgD. It is also monomer. Okay. IgG and IgD. These are the though this is the less quantity, very less quantity, 0.2% in the serum. But IgG and IgD, they are the most consistent one in the blood for the beta cell activation. What is beta cell activation? Whenever there is an antigen in the blood, there is an antigen, it enters the circulation. First cell to attack the antigen are neutrophils and what kind of cells? অনেক পরে আসে নিউট্রোফিল অ্যাক্টিভেশন অনেক পরে নিউট্রোফিলকে অ্যাক্টিভেট করতে হয় নিউট্রোফিল ইজ হোয়াট ইটস আ লিম্ফোসাইট the lymphocyte neutrophil বলি neutrophil is what neutrophil is a lymphocyte it's a lymphocyte okay so it takes time complement pathway দিয়ে it gets activated okay first cells are NK cells natural killer cells after that T cells. Then comes the B cell. So IgD helps in the beta cell activation. Okay. And acts as a receptor binding site for antigens. Some IgD what they does, they take up the antigen from the blood and they present it to the phagocytes. Okay. Phagocytes means the first cells, which engulf the antigen and kill. Exactly. Phagocyte কাছে নিয়ে গিয়ে এই IgD প্রেজেন্ট করে সেটা means and IgD antigenটাকে ক্যাচ করছে epitope paratope ম্যাচ হচ্ছে they are presenting it to the phagocyte will engulf the antigen বুঝে গেছো phagocyte will engulf the antigen This is IgE that has a very low concentration, but why does it increase IgE? Allergic reaction. Anybody who has a certain anaphylaxis or allergic rhinitis, we send them check for IgE. Okay. reaction. Means if there is an allergen is entering, it helps it. Basically, actually, it helps in type 3 hypersensitivity reaction. Okay, type 3 hypersensitivity. When we will study about hypersensitivity, I will tell you about it. Type 3 means already you got first exposed to antigen. Okay. Antigens polar entered to the first time before. Next time whenever you are going, the same polar is entering via your mucosa. IgE is getting activated. That's why people come with the complaint in OPD in your clinical rounds. You will get to see here. Medicine posting একটা দুটো হচ্ছে না, বাকি লোকেদের হচ্ছে তো। মেডিসিন পোস্টিং কোথায় হচ্ছে না? OPD তে চলে যাবে, এমনি তো মেডিসিন হয়। আমরা তো মেডিসিনের পেশেন্ট দেখি। অ্যাটেন্ড করবি। ওখানে you will get to see maximum patient will come with this complaint. সারা বছর কি হচ্ছে, হাঁচি কাশি বেড়ে গেল। কেন? Because the child is not exposed to one antigen, one colon, one A colon or anything. With that, their IgG level is to the roof, and another thing gets heightened up in case of allergic reactions. What is that? Eosinophils. Okay, eosinophil and IgE count, they gets very much heightened up in case of allergic reactions. Eosinophil. Acidic reaction is not a, but eosinophil count always get raised in an allergic reaction. Okay, parasite is also antigen, but parasite whenever that dies, that becomes more dead. Okay, parasite কিন্তু যতক্ষণ বেঁচে আছে এই লাইফ প্যারাসাইট থেকে, আমাদের গাটে প্রচুর রকমের প্যারাসাইট থাকে। তার মধ্যে সিঙ্গেল একটা যেমন গাট প্যারাসাইটের নাম বল, রাউন্ড প্যারাসাইট, রাউন্ড থাকে, বাট আমাদের এখন কেন এই রিঅ্যাকশনস কিছু হচ্ছে না। whenever they start multiplying, whenever they start dying, okay, whenever there is a hydrolysis, these things are very important. Last time few people of you are asking me in microbiology can you interesting লাগছে না, I told you to start from parasitic because it is very clinical oriented and it is very interesting. So you will get a very interesting with the subject. Start getting into the subject. Okay. It binds with the Fc receptor of the basophils and mast cells. I told IgE কি করছে, first reaction basophil কি করে, basophil and mast cells they have basophilic granules, they burst out and they give the allergic reactions. So IgE is important for the start allergic reactions. And substances like releases substances like histamines, vasoactive mediators means if you are going anaphylaxis from anything you are getting, you can get an anaphylaxis from a ant bite also. Some people they get anaphylaxis from peanut, anaphylactic reaction. All those are allergic reactions, sudden allergic reactions. So IgE is responsible for that kind of allergic reaction. And you have to, there is vasoactive mediator means there is sudden vasoconstriction. Okay, this kind of people they roam around with the pen to stop their, if they are having a cookies or anything without knowing which already has some kind of allergen, like if they are allergic to peanut, if they are allergic to egg, so they roam around with the anti-pen. What is anti? It is epinephrine or adrenaline. Okay, this is how. So this is the final thing. ছবি আমি আঁকতে পারি আমি জানি IgM যত মজার মজার হোনা, but this is the picture you have to write in your exam. You have to draw this pictures in your exam so you can take a